DNA replication is the process of copying DNA to make an entire new copy of your genome. So this is something that we're going to do every time our cells have to divide. So right before we're going to go do mitosis, our cells have to copy our DNA so that we have enough to make 2 new cells. So DNA is a double helix, which means we've got 2 strands of DNA and they're twisted amongst each other. So the very first thing we have to do is we have to open the DNA up in order to even get in there, to get into the helix, and this is going to create something called a replication fork. So here is our fork right here. And this is my helicase kind of coming in, it's moving this way, and as it goes, it's separating these 2 strands. The next thing we're going to do is complementary base pair. So each of these sides, we're going to have a protein called DNA polymerase, which sounds similar like RNA polymerase, but it's not the same. This time it's going to put down DNA, and it's going to base pair, and anytime it sees an A, it's going to put a T. And anytime it sees an A, it's going to put a T, and when it sees a G, it's going to put a C. When it sees a C, it's going to put a G, and when it sees a T, it's going to put an A. And so this happens on both sides. On one strand, we make our new strand this way. On this strand, we're going to make our new strands that way. But this doesn't happen in like a long continuous process. What actually happens is we make many short strands at one time, and then we have another protein here called DNA ligase, and ligate means to bind. So DNA ligase is going to come in here and connect these new strands together until we have 2 complete copies of the DNA. Semi-conservative, what semi-conservative means is that each of these new strands, right, so this is going to be a new strand and this is a new strand, have half of the original strand and half new, and then this side has half from the original strand, and then half of it is brand new. So let's take those 3 steps and kind of draw them out. So I have my double helix of DNA, and I'm going to use black to be the old strand, right? That's the original DNA. So the first step, right, number 1, is helicase, right? So helicase is going to come in and open up these strands. So we're going to end up going from these 2 things that are stuck together to having this kind of big replication fork. The second step was DNA polymerase. So polymerase is going to come in, and we'll go ahead and use blue to show the new strands. So DNA polymerase is going to go ahead and lay down some new strands of DNA. And then the third step we had to do was ligase, right? So what we're going to do here is now we're going to put all of those strands together and have 2 completely new strands. So if black was the old strand, blue is the new strand of DNA. So this is semi-conservative because for each strand, half of it is old and half of it is new. So it kind of conserves some of the old original DNA, and then you get your new DNA. Sometimes we make mistakes. So when we copy or when we do our base pairs, A's to T's and G's to C's, sometimes we mess this up. Think about it like typing up a paper. Have you ever typed a paper and made a spelling error or grammar error? Yes, you have, because I have too. But usually we have another molecule of DNA polymerase that can go through and proofread. So this is like spell check. It's going to go all the way down the DNA sequence and check for errors. Do I have every A and T together? Do I have every J and C together? Sometimes it's going to find these errors. And when it does, it fixes them. But have you ever turned in a paper where you made some spelling errors, but you did spell check and you caught them all, except your teacher still found a spelling error? That happens to me too. Right? So even though DNA polymerase goes through and proofreads for errors, it doesn't always get 100% of them. It does miss some. So sometimes mutations occur. So those are just changes, errors in DNA copying. Mutations can also happen from environmental effects, so things like UV radiation can come in and cause these two thymines to bind together. And look at the change in the shape of the DNA. When the DNA is shaped like this, it's not going to function properly. So here's the mutation at the sequence level, right? Here's my original sequence. It was supposed to be a CTG here, and then in the end we end up with CCG. And a mutation doesn't necessarily have to be good or bad. If this codon, right, so this is a codon, right? If this codon has no effect on the ultimate protein, then this mutation's just going to be neutral. It's not going to show any change. If it causes a change to the ultimate protein and it makes it a better protein, then this mutation might actually be beneficial. But most of the time what's going to happen is it's going to really mess up the protein and ultimately end up being detrimental. So a mutation is not inherently bad. It's the effect that the change in the DNA has on the protein that may be good, bad, or neutral. So take a minute and go ahead and reflect on the process of DNA replication in your own words.